DNA, also known as deoxyribonucleic acid, is a type of molecule that contains the genetic instructions that make us who we are. Most living organisms have DNA, and we inherit traits from our parents through DNA. At the Fred Hutchinson Cancer Research Center, scientists work on preventing early detections of cancer and other diseases. From the support of the Hutchinson Research Center and other funders, we got an opportunity to take part in a hands-on laboratory experience in our classroom.
On the first day, we learned how to properly set and prepare micropipettes, a type of tool that scientists use to help them make precise measurements.
The micropipettes are measured in microliters. 1 microliter is a millionth of a liter.
There are 4 different micropipettes, the P10, P20, P200, and P1000. To adjust the volume of the pipette, rotate the black volume adjustment knob. 3 of the 4 micropipettes have decimal points, and the 3 are P10, P220, and P1000.
The P10 has a range from 0.5 to 10 microliters. The P20 has a range from 2 to 20 microliters. The P200 has a range from 20 to 200 microliters. The P1000 has a range from 200 to 1000 microliters. Each micropipette uses a different tip for collecting dye samples.
In Thursday's experiment, we learned how to fill the wells with colored dyes and observe them as they traveled across the gel.
Electrophoresis is a process that separates charged molecules. Through the process of electrophoresis, the dye traveled from the wells of the gel to the anode or the cathode ends.
To prepare for the Agarose Gel, we will be using centrogram balance, weigh boats or paper, a bottle or flask with 3 times volume of gel solution, graduated cylinder for Agarose solution, a microwave oven or hot plate, 150 milliliters of plastic beaker to hold the buffer, Agarose powder, and 1 TAE buffer.
In this experiment, we will be using electrophoresis apparatus, 1% Agarose Gel, micropipettes, ruler, power supply, 1 TAE buffer, microtube rack, breaker for use tips, micropipette tips, marker pen, microtubes, and a centrifuge.
Step 1. The gel is made by dissolving agarose powder in a buffer solution by microwaving the solution. After microwaving it, stir the solution for about 2 minutes, then pour in the solution. Step 2. Add 1 TAE buffer to just cover the gel about 1 through 2 millimeters. Use dams to hold the buffer in place and insert the comb in the center position. Wait for it to solidify before adding your samples.
Step 3. To prepare your samples, number and label the tubes with the names of the samples you have chosen. Step 4. Collect the samples by pipetting 20 microliters of each sample from the class supply into the appropriately labeled tube. Step 5. Before loading samples, be sure your gel box is near the power supply. Load 15 microliters of the sample into the appropriate gel well.
Step 6. When all 8 samples are filled, close the lid of the gel box. Each group will share the power supplies. Connect the electrodes to the gel box and the power supply connecting red to anode and black to cathode. Turn on the power supply and set it to 100 volts. Check the milliamp output. Electrophoresis for a total of 10 minutes. Step 7. Observe the changes in the gel box after 5 minutes. Step 8. After 10 minutes, turn off the power, unplug the electrodes and open the gel box. Record your results in the gel diagrams.
On Friday, we did another experiment with electrophoresis, this time staining the gel after it was cut by restriction enzymes.
Restriction enzymes are proteins used to cut DNA at specific DNA sequences.
In this experiment, we will be using 1% Agarose Gel, micropipettes, electrophoresis apparatus, power supply, 0.25 TAE buffer, ruler, beaker for use tips, micropipette tip, microtubes, centrifuge, 100x fast blast, staining tray, and light box. Step 1. Prepare 0.7% Agarose Gel. Step 2. Prepare the samples. Label the tubes 1, 2, 3, and control, which is C. Start by putting in the salt buffer into each tube. then DNA/E. coli, and so on. Add sample loading buffer last. Don't forget to change the micropipette tips every time you use a different sample. Step 3. Mix gently, then place them in the centrifuge for 3 seconds. Make sure your tubes are balanced.
Step 4. After the gel is set, take out the comb that is near the negative electrode and the dams. Add 1x buffer so it creates a light layer on top of the gel. Step 5. Now take your samples and load 15 microliters of each sample into the wells. Close the lid on the gel box. Attach the electrodes of the gel box to the power supply. After this, electrophoresis it for about 5 to 10 minutes, or until the bromophenol blue runs 2/3 of the way through the gel.
Step 6. After 20 minutes, turn off the power supply, then record the new placement of each dye on your record sheet. Place the gel into the staining tray and start the making and stain gels with fast blast steps. Step 7. Once you have finished destaining your gel, record your results where the bands traveled from the wells.
Step 1. Stain. Once the electrophoresis process is done, place the gel into a staining tray. Pour the 100x fast blast into the tray covering the gel. Let it sit for 3 minutes. When done, pour the stain back into the fast blast container.
Step 2. Destain. Now start to rinse the gel with warm water while gently moving it for 5 minutes. Then discard the water and look at the final results with the light box.
We learned how to calculate the cuts that are made in DNA after the process of electrophoresis. Each student was given a photo of a DNA restriction map that they had to use to write down the cuts made by E. coli and HIN3. After that, we had to combine E. coli and HIN3 into one line and find how many cuts it made. The sum of the cuts by E. coli and HIN3 had to be 48,502.
This project has given us a glimpse of how scientists research in their lab and how much work and dedication goes into these researches. Thank you, Fred Hutchinson Cancer Research Center, for donating materials for the sake of learning.